So if you look at these, the content inside the phloem sieve tube, we call them as the phloem sacs. Okay, the phloem sac. So if you analyze this using this castor oil plant, so I will explain why we use the castor oil plants. Because not all the plants we can analyze them. So if you analyze the composition, we can use the castor oil plant. Okay, so if you analyze it, you realize that in this table, few things very interesting. Sucrose, particularly in very, very high concentration. How high? Now, if you understand, this is mole per dm3, the concentration, 250 mole per dm3. If you still remember the concentrations of the sucrose that we use for our experiment. Still remember last week, we used 1 molar, also 1.5 molar. The highest we use is 1.5 molar sucrose. Okay, last week. Okay, but this one is 250, it's more than 100-fold of what we use. So means that photosynthesis take place, photosynthesis in the plants, the process actually converts the glucose into the sucrose and to be transported within this xylem, within this phloem sieve tube element. So that's why the concentration is very, very, very high. Okay, so next you can see that amino acid as well is very, very high, and other concentrations also you can see that. Are you clear? But one thing that we a bit like, you have to understand here, 2 items I want you guys to highlight, ATP and also the nitrate ions. Okay, so what's ATP? ATP, because we do know, why I circle out or highlight the ATP, because we know that ATP will be produced, okay, within the cells. So means that the present ATP actually also indicates that sieve tube elements, they do have their own mitochondria. Okay, they do have their own mitochondria. But later stage, when we talk about this mitochondria, then we will see that, okay, we will see that this seems like no functions inside the sieve tube element. And this ATP also can move from the companion cells to sieve tube element also, okay, through the plasmodesmata. So that's why you can see that we do have the ATP, okay, in this phloem sac. Nitrate ion special. Okay, why is it special? Because you can see that the concentration equal to 0. So you can write down nitrate, transportation of nitrate, always in xylem. Okay, nitrate will be used. Why is for function so important for nitrate? Nitrate, we know that for the protein, we need nitrate. DNA, RNA, all need nitrate. So means the plant, we're going to use them up. Okay, when the xylem transport this nitrate from the root to the shoot. So therefore, nitrate won't be transported in these phloem sieve tube elements. Okay, so in order to collect the sufficient liquid, okay, to analyze or phloem sac analyze, we have 2 ways actually. So we can actually use the castor oil plants, or we use aphid. Now, why I purposely highlight these castor oil plants? Because if you look at these sieve tube elements, similar to the lignified xylem vessel elements, they are under high pressure. Okay, so if they're under high pressure, okay, if you cut it, if this is the plant's sieve tube element, when we start to cut it, what will happen here is they were going to have something like a leakage of the fluid. So this leakage will cause, okay, the release of the pressure. Are you clear? So release of the pressure inside cause a surge of the content, and plant actually responded by a proper seal with a carbohydrate, no callus. It's something like clotting mechanism, blood clots. So it's something like, oh, leakage already. So plants start to seal it. So it means that if you use most of the type of plant, most type of plant actually do have this clotting mechanism. Right? Means that they do have the ability to clot the sieve pore, block the sieve pore, so you won't be able to collect a lot. So you look at this diagram, we do have the short-term protections and also long-term. Short-term, they use pea protein, phloem protein. So pea protein here, can you see I start to block. Okay, then after we put a short-term protection, we want to have the permanent protection. Then callus is the one that actually will be synthesized. Callus, a type of carbohydrate, will synthesize and block. So once it's blocked, then these safety elements no longer can be used. Are you clear? So therefore, that's why we won't be able to collect sufficient fluids, okay, to be analyzed. Okay, so how we can do this? How we can do this? So we can use this castor oil plant. Why? Because the phloem sap will still continue to flow from the card for a longer period of times compared to other plants. Means that they don't have a proper sealing mechanism to seal the sieve pore. So allow us to check. Okay? But the question here is, we also want to compare the phloem sap of the castor oil plant with other plants. Correct or not? So how? So we can use the aphids. So what we do, because aphids, they rely on the phloem sap. So these aphids, the insects, they do have this something like the mouthpiece known as the stylet. So this stylet will know where to puncture, where to go to get the phloem sap. So we just let them, okay, let these aphids go to the plants, and then they will help us to find. Okay, so the aphid will insert their stylet through the surface of the plant stem or the leaf into the phloem. So the phloem sap will flow through the stylet into the aphid. This is by the pressure. We know that the sieve tube elements is under pressure. So under pressure basically means that if you poke a hole, this liquid will come out. So means that very simple. This aphid do not need to suck this, okay, or withdraw the fluid or this phloem sap, because they themselves automatically the phloem sap is under pressure, it will go and leak out. Okay, so what we're going to do here is, if you look at this diagram, so can I see the mouth, the mouthpiece? So we're going to freeze, okay, use the CO2, okay, I mean to freeze, okay, the CO2 to freeze. Once we freeze these aphids, what we can do, actually we can cut at the mouthpiece here, remove these aphids. You can see that the liquid actually flow out. Can you see that? Why? We do not need to pump it out. Why? Because this also shows that our xylem, as well as phloem sieve tube elements under pressure. So when under pressure means that this pressure will force the liquid out. And because of the stylet is so thin, it won't trigger the sealing response from the plant. So the plants won't, okay? So the plants won't actually, how do I say that? Seal it, because it's too thin in this case. Okay, so too thin, too small, the plant actually didn't realize it. So what we can do, we actually can, okay, take this. You may see that, oh, so small number, so small, but we don't use one aphid, we use many aphids to help us to collect this the phloem sap. Okay, then we can do the analysis. What are the content inside this phloem sap? Okay, so with this, I've done for the parts. Okay, the contents of the phloem sieve.